The story goes that one day they saw a white-eyed fly flying around in one of the tubes. And this is unusual because normally flies have red eyes. So normally it's red eyes, and the white-eyed must be some kind of mutant. Well, of course, being a geneticist, what you do is you take the white-eyed fly and you cross it with a red-eyed fly, just to see what happens. And so that's what they did. And let's look at the crosses here. So here is a red-eyed female fly, and the crosses, you can see the two red-eyed genes on the chromosome. And we have a white-eyed male, and you can see the white-eyed gene here. And when you cross those together, here's a Punnett square. You can see that this chromosome goes here, this chromosome goes here, and because these two are the same, because she's homozygous, then there's only one possibility, and that goes here. And so here are the progeny of that first cross. We call it, of course, the F1 project cross. And we see that the females are heterozygous. They have red and white alleles, but of course, because red is dominant, we have a red-eyed female. Where we have this chromosome with this chromosome, this X and this Y, we also find that the male has red eyes because it has one red allele. Now it is hemizygous because it only has one allele. There's nothing on the Y chromosome here. And so these white and red eye alleles are on the X chromosome. Now Morgan didn't know this at the time. He just thought they were just standard autosomes. But he's now going to find that they are actually on the X chromosome. So if we go over here to the second cross, this is the same kind of thing where we have a red-eyed cross with a white eye, except that the genders are reversed. Here we have a white-eyed female. You can see here she's got two recessive alleles, meaning white eyes, and here's a red-eyed male. So it's still hemizygous, but this is the dominant trait. If we cross these two together, there's the Y chromosome, here's the male X chromosome, and because the two females chromosomes are the same, we just put one in here. We find that the new females that are made are red eyes and are heterozygous. Here's the dominant one and here's the recessive one. But look at the males. We have white-eyed males because they inherited the white allele from the female and of course the Y chromosome from the male, so they have white eyes. So this was one of the strange things that Morgan found when he did these two crosses, and they are called reciprocal crosses. Reciprocal crosses. They're reciprocal because one is the reverse of the other. So here is a red-eyed female crossed with a white-eyed male, and this one is a white-eyed female crossed with a red-eyed male. It's the opposite, it's reciprocal. And look at the results of the F1 generation. F1 on the first case, all the progeny are red. F1 on the second case, they're half red, half white. And so this was very unusual for Morgan. This was unexpected. And so he went on to explore this further, and if you actually take each of these F1 progeny, take them through to the F2 stage, and we won't do this on here, we'll do this in class, you see that half the F2s were white eyes in the right-hand case here, and only a quarter of the F2s are white-eyed in the case on the left. And so you should be able to follow this through in the parental generation F1 and F2, and we will go over this in class to make sure that that's okay. So now we know how sex chromosomes are inherited. Let's look at the human sex chromosomes, the X and the Y chromosome. And what we have here is an electron micrograph of human sex chromosomes. This one on the left is the X, and this over here on the right is the Y chromosome. And you can see there's a huge difference in size. The Y chromosome is significantly shorter than the X chromosome. And some recessively inherited X-linked traits. Now these are recessively inherited. So the way we write this is, here's an X chromosome, and if it's the recessive trait, we'll put a little a as a superscript. If, shall we say, it's a dominant trait, we'll put a capital A, and A on the X chromosome. And so these are X-linked recessive traits, and there's a number of them that you may have noticed. Here's color blindness up here. Here is hemophilia, which you've probably heard of, and here's muscular dystrophy as well. There are many traits on the X chromosome, and they're called X-linked, of course, because they're on the sex chromosome and the trait are linked. That's the same molecule of DNA. The gene, the A gene, the A allele, is on this X chromosome. So here is a X-linked recessive inheritance pedigree chart. So let's start with let's start with number 5 down here in this last generation. We'll make this the proband. And this is a recessive trait. We know it's male, so it has to be XY. It's a recessive trait, so the gene on the X chromosome is little a. So the genotype of this individual number 5 is X superscript little a Y. So if we follow this back, we can work out the genotypes of the parents. Of course, mother has to be XX, father has to be XY. The Y chromosome went from the father to this son, so this X superscript little a must have come from the mother. So we know that she has a little a on one of her X chromosomes. But the mother herself does not display the trait. Therefore, she is not double recessive. So her other X chromosome must be X superscript capital A, or uppercase A. So mother is heterozygous for this A trait. Father does not show the trait, so he must not have a recessive trait, so he is X superscript big A. So there we can work out those two parents. And of course now we can work out the other children as well. These are females, so they must have 2 X chromosomes, and if they do not have the trait, then one of those X chromosomes must be dominant A, that came from the father, and the other X chromosome could be dominant A from the mother or recessive A from the mother, in which case they would be carriers. We just don't know what they are here at the moment. Another example of X-linked recessive trait is color blindness. Detection of color is due to specific proteins in the retina. If these particular proteins that detect either red, green or blue light, if they're mutated and they don't work, and then you don't detect color. And so there are 3 different forms. There's this red, green and blue proteins, and either one of these could be mutated, or a combination could be mutated. So how does this actually work? Here's a diagram of the eye, and the back membrane of the eye is the retina, and here's a cartoon of the different cells of the retina. If you remember your eye physiology, these are cone cells, C-O-N-E, and there are different types of cone cells. There's ones that detect red light, blue light and green light, and the other cells are rod cells, and they detect just a light intensity, black and white effectively. You can test for color blindness. Here is a classical colored dot marker, and it's a series of dots of different shades, and within those dots are numbers, and you're supposed to be able to tell which number it is, and whether you can see something here, but there is something around here. You can maybe just about make that out. If you go to this website, that has the full test, and you can test yourself to see if you're colorblind or not. And if so, where did you get your colorblind genes from? Was it from your mother or your father? We'll ask that question in class as well. On a more serious note, another recessive X-linked trait is muscular dystrophy. And this is basically degeneration of muscle cells. It's a gene on the X chromosome, so it's X-linked. And here's the biochemistry behind it. This is a cell membrane, right here. And that cell membrane in a muscle cell is connected to, via various proteins, to this molecule called actin. which is down here. And actin proteins are essentially muscle proteins. They can contract and they give you muscle function. This protein called dystrophin connects the actin muscle fiber to the proteins in the cell membrane. If this dystrophin protein does not work, then the actin fiber will contract and it will just rupture the cell membrane, and thus the cell will die. And so what will happen, you have a single gene producing this dystrophin protein, and if that single gene on the X chromosome is defective, you don't get this dystrophin protein. If that's the case, the actin muscle fiber is not connected to the membrane, and the membrane will rip, and the cell will die, and thus you have muscle atrophy. So here is another X-linked trait. It's an X-linked recessive gene. This is for hemophilia, and this is famous for Queen Victoria being the originator of a line of hemophiliacs throughout the European royal families back in the 1800s. Here's Queen Victoria. She is a carrier, and so she has X with a superscript big H and X with a superscript little h, because she's a carrier for hemophilia. Prince Albert, her husband, is X superscript big H because he does not have the disease, and Y, so he's hemizygous dominant. Victoria then does not have the trait. She's XX for being a female, and she has the dominant from her father and the dominant from her mother. King Edward, male, but he has the dominant from the mother and the Y chromosome from his father. Now Alice here is a carrier, so she is X big H, and she got that from her father, and X little h, and she got that from her mother. Beatrice is also a carrier in the same way, and Leopold actually has the trait. So he has X little h and the Y. The Y obviously came from his father, Prince Albert, and the X little h came from his mother. So we have currently talked about X chromosomes, but of course there's also a Y chromosome and the sex chromosomes, and you can have inheritance patterns with Y chromosomes. And so only males, of course, have Y chromosomes, and the genes on the Y chromosomes are passed directly from the father to the son. And of course we only have one Y chromosome, and so we are hemizygous for genes on that chromosome. So here's a cartoon of the Y chromosome. The PARs up here, that stands for the pseudo-autosomal region. There's one at either end. And what these PARs are for is for pairing with the X chromosome during meiosis. So if you remember, during meiosis, all similar chromosomes line up and they can cross over, or they pair together. And so you might get two sets of chromosomes lining up to each other and separating during, say, anaphase 1. Well, of course, the X and the Y chromosomes are totally different, but they have a little bit of DNA that's similar so that they can line up during this process. So there may be an X chromosome lined up next to a Y chromosome lined up during anaphase, and they can be separated in that region. And here are the PARs just at the very end of the Y chromosome, and that can pair up to the X chromosome. The other important gene up here is called SRY, and SRY stands for the sex-determining region of the Y chromosome. And it's important, at least to males, because if you have SRY, then you are male. That is kind of the male trigger for development. If you have SRY gene, you end up being a male. There are also such important genes on the X chromosome. There's a gene called SXL, stands for sex lethal. Its function is an RNA binding protein, and if SXL is on, then you end up being female. So this is a determination of sex on the X chromosome. On the Y chromosomes, SRY, which we just talked about, and it is a transcription factor.